We'll get started. All right, welcome to the afternoon sessions. We've got three tracks going today, and 3 sessions this afternoon on each channel. I guess we do have one that had to pull out. So at 2 o'clock, there's only 2 choices now. But I want to introduce Eileen. She is going to be giving us an introduction for Stantec. Welcome,
Hi,
Eileen.
guys. So good afternoon, everyone. As Emily just said, my name is Eileen Weigel, and I'm a water resource engineer at Stantec, the host of this session. Since you are not attending this conference to hear about Stantec, I am going to simply skip our intro. I will put my contact information in our chat session, and we'll have a representative at the sip and share this afternoon if you do have questions for us. At this time, what I would like to do is thank Coon Creek Watershed District on being an excellent client and a continued thought partner on research ideas to further advance protection of our water resources. It's been very enjoyable to work with not only the individuals that are presenting here today, but many of their other coworkers. So without further ado, I would like to introduce this afternoon's session entitled A Tale of Two Filters, Amending Filtration Media with Biochar to Remove Bacteria from Urban Watershed, presented by Coon Creek Watershed District staff, Justine, Dauphinais, Water Quality Coordinator, and Chase Vanderbilt, Water Quality Specialist, both who happen to be showcased on the best picture video during lunch, sampling these very systems we're about.
Great, thank you, Eileen. And the feeling is mutual. We have been enjoying working with WENC, now Stantec. So thank you for that introduction. I'm going to go ahead and attempt to share my screen here. So let me know how it looks.
We can see your screen.
Are you seeing full?
Now we're seeing the
note or the notes.
We're not seeing notes, and we are seeing just the one slide. So I think You're exactly where you want to be.
I think you're right. All right. Well, then, like Eileen said, my name is Justine Dauphinais, and here with me today is Chase Vanderbilt. We're both in the water quality program at the Coon Creek Watershed District. Before we jump into the talk, I want to acknowledge some co-authors on this work. John Janke in our office. He's our operations and maintenance coordinator, and he was the project manager for construction of both filters. And then I wanted to acknowledge our design and engineering team, both Eric Migau and Ed Mathieson of Stantec. Also wanted to acknowledge our municipal partners, the cities of Blaine and the city of Coon Rapids. They both played a role financially in these projects, as well as signed on for long-term maintenance. So that was very helpful. And then these projects could not have been completed without funding, in large part from 2 competitive grants from the Clean Water Fund administered by Bowser. So with that, today we're going to talk a little bit about the problem, excess bacteria in urban stormwater, and then introduce one of our proposed solutions using biochar as a filter media amendment. And then I will turn it over to Chase to introduce our specific filtration BMPs and go over our performance monitoring plan. And then I'll take it back over to share with you our first 2 years of preliminary performance results and tie it all together with some conclusions. So with that, the problem, excess E. coli in our urban stormwater. So E. coli is used as an indicator of potential human health risks. And I wanted to bold and underline indicator just to stress that having E. coli in your water does not mean you have human health risk, but it is indicative of possible sewage contamination, and it's correlated with with prevalence of other pathogenic bacteria and viruses. And these specific pathogenic bacteria and viruses are much harder to measure and very expensive, whereas E. coli is relatively simple to measure, repeatable, and economical. So that's why E. coli is used as an indicator, and it is the basis for recreational use impairments under the Clean Water Act, nationally and in Minnesota. So when it comes to recreation impairments, in Minnesota, we have almost 900 water bodies that are impaired for recreation due to excess bacteria. E. coli specifically, or formerly, some stretches of river were impaired under fecal coliform contamination. And in the metro area alone, we have just shy of 100 of these impairments as shown on this map. And if you zoom into the Coon Creek Watershed District, which is located here in central Anoka County, all 4 of our major streams are impaired for aquatic life due to excess levels of E. coli. And we have a TMDL, a total maximum load study that quantified our existing levels of pollution, where we need to be in order to meet these standards for safe recreation. And it has been determined that we need to reduce our E. coli levels between 30 and 70% in these 4 different streams. So it's not an unattainable 99% reduction that we need here, but it is pretty sizable, and it's going to take a lot of different strategies and a lot of time to accomplish. So whether or not you agree with using E. coli as an indicator of health risk, there are some problems, but you can't deny that E. coli and excess bacteria is causing real problems locally in Minnesota. So I just have a couple snapshots of a few news articles from the last couple of years. So in 2019, we had record levels of rain, and you can see there was some series of press that came out about beaches being closed because of high rain and high E. coli. And even in Lake Nokomis, there were 73 confirmed swimmers that were sick with an E. coli outbreak that was tied back to Lake Nokomis. And then this year, where we had drought and a warm stretch, I thought this article was interesting. And high levels of E. coli won't stop beachgoers amid a hot streak. And it goes on to talk about Bidé-Makaska and Cedar Island Lake, and how there was prolonged beach closures. Yet after the first couple of days, there were hundreds of families and kids swimming every single day. So I don't think we can deny we have a problem in urban areas with bacteria in our stormwater. And in order to figure out how we might go about addressing this, we really have to understand the sources. So broadly, there are several anthropogenic sources, human sewage specifically, depending if you're in a sewered area or not. That would be septic systems or leaky sanitary pipes, or in some cases, sewer overflows. There's also domestic pets, namely our dogs. And then more recently, there's been kind of a body of literature looking at regrowth, resuspension of E. coli kind of outside of the animal gut in our stormwater infrastructure, in our curb lines and gutters and catch basins and sumps and vaults, where we have nutrient-rich, muddy, organic material that's wet, and it's just fosters bacteria growth and regrowth, and then can be suspended and sent down to our receiving waters during storm events. There are also several natural sources of E. coli, and I have natural in quotations here because a lot of these can be kind of exacerbated by humans in urban areas. For example, waterfowl. Of course, they're eating and pooping in our waters, but in areas where we have aeration or manicured lawns along stormwater ponds, or where we're feeding waterfowl, you get kind of unnatural aggregations that can really be E. coli loading hotspots. And then there's other natural sources, of course, deer, aquatic mammals, beaver, raccoon, things like that. And then there are natural environmental reservoirs that also regrow and resuspend E. coli, things like our stream bank and our stream bed and just soil and plant material. And these can too be exacerbated by humans. Stream banks, for example, it's okay that you have E. coli living in that soil, but if you're in a place that has altered hydrology and excess erosion, then that's getting resuspended and added to your receiving waters at a much higher rate. So kind of one commonality when it's looking at E. coli in urban systems is a lot of these sources are more non-point source. They're diffuse in nature versus outstate, where we have a lot of agricultural things like feedlots and manure lagoons that are more point source and maybe easier to manage. So when it comes to addressing and reducing E. coli, first and foremost, we're looking at source reduction strategies, things like picking up pet waste and proper care of organic material, leaf litter, not irrigating as much, and all of the education and outreach that goes along with this. But what we also want to look at is how do we reduce this E. coli from all of these diffuse sources after they already enter our receiving waters? And that is where biochar comes into play. So what is biochar? This is showing a picture, a handful of the actual biochar we used in our filters here. It is a charcoal-like substance, and it's made via pyrolysis of organic material. It can be all different sorts of organic material, trees or crop residue like peanut shells or corn husk or even manure. And then you burn it at a high temperature in low or no oxygen. And it turns into this really great carbon source that it's not a new product, new technology. It's been around for hundreds, thousands of years, really. And it's used as a soil amendment in agriculture. It helps add moisture and soil structure and helps grow plants. But more recently, it's been gaining a lot of traction as a potential amendment for filtration media for stormwater. And it is a promising filtration media because it has immense surface area and a really complex pore structure, really tiny pores, some bigger pores, some different kind of functional groups that have different binding properties. It has proven absorption of heavy metals. It's been used a lot in remediation of like known contaminated sites and groundwater. And then more recently, it's been shown to also remove bacteria, specifically E. coli, in simulated stormwater in lab column trials. Very, very effectively, over 99% removal. And then we kind of use this lab data and working with Stantec and some other water management organizations in the North Metro have embarked on a series of really small scale field trials over the last 5 years. And we saw promising results moving from the lab to the field on the order of 50 to 90% removal. So based on this, we were ready to kind of scale it up and build a real full-size regional BMP. But first, we had to figure out what biochar we should use. And it's really important point that not all biochar is equal. There's countless different varieties depending on what organic material was used and what temperature it was burned at, or if there was any post-processing occurring. So we set out, we partnered with the Mohanty lab at UCLA, and we found 4 commercially available biochars shown here in this graph. And we sent them to the lab for column testings to specifically look at their ability to remove E. coli bacteria, since that was our kind of target pollutant. And all 4 of these honestly did really, really well at removing E. coli. And our worst one was 97.8% removal, and our best was 99.9. So we went with the best one, and it also happened to have an office in Minnesota, so it was convenient as well. And that was called Agricultural Carbon, and it's marketed by a company called National Carbon Technologies. They're out of Oakdale. And our source material was hardwood. It was burned at 550 degrees Celsius. And then we did a whole slew of testing just to kind of characterize its characteristics, I guess. The surface area was incredibly large, 339 square meters per gram, which I know is an obscure number. So we kind of did the math. And if you were to lay the amount of biochar we used, lay that surface area out, it would cover the northern half of the 7-county metro area. So immense surface area. And then our particular biochar, this is the composition. Again, it's probably kind of meaningless to most of you, but working with the group at UCLA, they actually published a paper based on these lab bench trials, looking at the various kind of contents of biochar and they modeled how it would interact with bacteria. And they found that the biochars that had the highest surface area and the most fixed carbon content did the best in physically entraining and absorbing E. coli. And if you had too many impurities, namely volatile matter or ash, these performed less well because it would limit absorption or repel negatively charged bacteria. So just kind of interesting. So if anyone's looking at biochar, there's a lot of intricacies that goes into picking your biochar and making sure it's good for the pollutant you're targeting. So with that, I will turn it over to Chase to talk about our specific filters.
Great, thanks, Justine. So as Justine mentioned, we scaled this up to full-size filters. And so that includes 2 filters that we have online right now. And both of these filter BMPs are comprised of 2 identical iron-enhanced sand filter cells, but also one of those cells has biochar added. So you'll see in the rest of this presentation, IESF versus BIESF. Well, the B is just denoting which is the biochar side of the given filter that we're talking about. So first, our woodcrest filter. This is a gravity-fed pond bench filter with about a square mile drainage area, and you can see that in the yellow off to the right there. And then the second filter is a pump-based filter, and that has a drainage area of just over a half of a square mile in dark red there. So first, our woodcrest filter, it's a very large surface area. We have 22,000 square foot surface area. You can see it's kind of outlined in the yellow there. And then down the middle of that is a yellow dashed line. That just shows the two different filter cells. And there's actually a berm on that line that separates them so they can be sampled individually. And in this case, the right side is the biochar side. And then below this filter in the picture, just to the south of it, is a flow-through pond, which has an outlet control structure. And that structure has a sluice gate in it. So when we close that sluice gate off almost all the way, that allows base flow to still pass, but the pond fills up just under the level that it would overtop into the filter cells. So it kind of rides that line until it rains, and then the pond, heads up as that happens, and then submerges the filter and filtrates through, and then goes through a drain tile system individually back to Woodcrest Creek. So here's just a picture of kind of an on-the-ground picture. It gives you a little bit of a scale reference. You can see that bobcat off to the right, kind of shows you how big this area actually is. And this is at the end of construction when they're actually tilling in the biochar. So you can see that's what that black substance is. And then off to the left is that flow through pond. Right now that sluice gate is open in this picture, so it's just allowing base flow to completely flow through. But and that's circling the outlet control structure there. And then when we close that sluice gate and get rain, this is what it looks like when the filter is fully operational. So this is a day after an inch rain event and this is obviously fully submerged, fully operational. Everything is working as it is designed to. Switching over to our Pleasure Creek filter. So this is our pump-based filter. And off to the right, there's a large stormwater pond that this draws water from. And then there's a pump valve here that disperses the water into the two filter cells. And we can set it however we want, but it goes back and forth between the cells. And we typically do 8 to 12 hour time periods on each cell. So it switches back and forth. And then in this picture, it's pretty obvious. The left side is the side with biochar added, and the right side is the iron enhanced sand only side. And then you can see those 3 rock piles. Those are actually covering the distribution heads. So when the pump is on, water cascades out through those and then evenly disperses over the filter bed, filtrates through, goes through a drain tile system, and then gets put back under the road back to Pleasure Creek on the other side in a cleaner state. And then for our performance monitoring, so we take paired grab samples of the untreated water and then versus the filtered effluent on both filter cells individually. So we send those samples off to the lab to get analyzed for E. coli, total phosphorus, orthophosphorus, and total suspended solids. For each of these samples individually, we also take a SON measurement, just in case we need to go back and say there was phosphorus leaching or something, or an issue that arises. We have sort of a record of physical conditions of the water at that time, so we might have something to point at if oxygen is low or something like that. We kind of have this as an insurance policy. Also for our treated volume calculations and our pollutant reduction, load reductions, we have continuous flow measurements at both of these sites. So at that Woodcrest site, the gravity fed filter, we have area velocity sensors in the outflow pipes. And then at the Pleasure Creek filter, which is the pump-based filter, we just have a pump rate integrated, so that records automatically for us and can be downloaded at any time really easily. And then finally, we also have continuous level loggers in all of our media beds. And this helps us if we're running into any issues with clogging of the media, or we just want to track infiltration rates over time and see if they're changing. And then also at the Woodcrest site, we have a level logger in the main pond, which is also connected to a telemetry unit. So real time, I can check, if I know rain is coming, I can then sign in and check if that water level is above the filter media at any given time. And we'll kind of show you that in the next slide here. So getting into the event sample that we'll show you results for, or some of our results for. We got a little bit of a late start. We wanted these filters to be online earlier in the season in 2020, but supply chain things were not the greatest at the time. So we started a little late, but even at that, we still got 8 events sampled at Woodcrest, ranging from 1/10 of an inch storms all the way up to just over an inch. So you can see in that graphic off to the right, the rain events are denoted in the green bars, the secondary y-axis, and then that continuous blue line is the actual pond level throughout the season. And then that red bar that kind of cuts this in half is the elevation that the main berm separates the filter cells from the main pond. So if you see that blue line is above it, that means the filter was in operation at that time, which kind of leads me to, you can see all of the events that we sampled, that we took grab samples, are shown with those yellow diamonds. And we did take just grab samples at all these, like I said, and we did use those to calculate event mean concentrations, which isn't necessarily ideal. We would love to have an auto sampler connected to these systems so we could sample the entire hydrograph, get better mean concentrations. But working with sampling for E. coli, that gets complicated because, as Justine said, E. coli can actually reproduce in sewer systems and things like that. So all these pump tubings and things that are involved in auto samplers could actually host bacteria. So our goal when we have time and resources is to just physically go out 5 times over a storm and capture that in the future. And then finally, at the Pleasure Creek filter, this is really easy to sample. It's always pumping. So we were able to get 10 samples really easily that year on a weekly or bi-weekly basis, depending on how many we want. And so that's really easy for us to do. I think with that, Justine will get into some of the results.
Yeah, thank you, Chase. So I'm going to go over all of the 2020 results first, and then I'll go over the 2021 results after that. And I'm going to show you a series of graphs. So first, I just wanted to bring up one and kind of talk through all the elements, and then there'll be a bunch of them in the same format repeating. So what we're looking at here is the 2020 untreated, the influent concentration versus the filtered effluent concentration for just E. coli at just the Woodcrest filter. And we do have the 2 filter cells. So the dark blue bars, this is untreated influent. And then the gray and the green, these are the 2 cells. The gray, biochar added. Gray, you can think charcoal. And then the green is just the iron enhanced cell without the biochar. So a few things here. This is just concentration. It's just on a normal scale, not a log scale. And you can see we have these asterisks here. And that is because E. coli has a, if you submit it to the lab, there's kind of a maximum reporting limit of 2260 is basically like how much E. coli they can count. And they just say, well, it's above that. So we ran into that a couple of times. So all of our estimates are actually conservative because we know there was at least 2,200 colonies of E. coli, but probably more. So midway through the year, after getting a couple of those samples, we actually started taking replicate samples and then sending them in for dilutions so we could up that kind of maximum reporting limit. So then that's why over here towards the end of the year, we actually could enumerate E. coli, you know, up to 3,700 here. Couple other things about this graph, you can see some horizontal lines. So E. coli has 2 water quality standards when assessing systems for recreational impairments. This yellow dotted line, this is at 126, and that is the chronic standard, that the long-term average of your samples have to be below that to be supportive of safe recreation. And then there's an acute standard at 1260, where only 10% of your samples can exceed that. Otherwise, you're not supportive of recreation. So those are indicated here. So what you can see is our untreated water, these dark blue lines, are generally much higher than our filtered water. So that's a good sign. And some of our untreated water is quite high, you know, way above that acute standard. And even the filtered water has dropped down to more acceptable levels. And you can see in most of these events, these are just in chronological order, that the E. coli level is a bit lower in the cell that has biochar, showing that biochar is outperforming just the iron enhanced sand side. This is true in most of the samples, but there are a couple here. You can see where the filter without biochar actually had lower concentrations. But interestingly, these are in the 2 smallest events. So it seems like the biochar is working better when there's higher concentrations, kind of on average. So I'm going to show you the same thing for the Pleasure Creek filter. This is the same exact access. I did that intentionally, not to annoy you because you can't really make out what's happening, but just to show that there are much, much lower incoming levels of E. coli at the system. So low, in fact, that trying to look at percent reductions is kind of meaningless when you have, you know, the difference between 50 and 20 or 5 and 2. Those percent reductions get kind of skewed. But we did have a couple events here where we exceeded the chronic standard and one where we exceeded that acute standard. And you can see that the filtered water is much cleaner than that incoming water. So I'm just going to show the same graphs now for some other pollutants. So when looking at total phosphorus at Woodcrest filter, same type of graph, but now for phosphorus, the state standard is 0.1 milligrams per liter. So you can see that incoming water often exceeds that standard, and our filtered water is generally pretty clear, pretty good, 0.02, I'd say on average. And here the green and the gray bars are quite similar to each other. There's not an obvious difference of kind of what type of filter media is outperforming the other. And kind of the same thing at Pleasure Creek. And again, here we just have much lower levels of incoming pollutants. So it's, you know, we're having removal, but when your incoming load is only 0.02, 0.03, your filtered water, and this is, you know, less than 0.01, it's very, very clean. Not a whole lot of phosphorus to reduce in the first place here. Looking at orthophosphorus, so this is the dissolved fraction. We looked at this separately because, as many of you are aware, I'm sure, the iron filings in iron enhanced sand are specifically designed to try to get at this fraction of phosphorus to bind that dissolved fraction and kind of entrain it, immobilize it. So we saw pretty good reduction here at the Woodcrest filter. Again, our incoming levels were more about 0.05 and the cleaner water was closer to 0.015. And then the same thing for Pleasure Creek filter, very, very low incoming loads, so much so that many of our vents where you don't see bars, those were actually non-detects. You can't detect phosphorus below about 0.003 in the lab, so very, very low. But here, interestingly, you can see the couple of times where it was detected. It was actually detected in our filtered water at a higher frequency and higher concentration than in our incoming water. So this does show a little bit of leaching of the dissolved fraction. And I'll talk about that in a little bit more when I show you guys all the loading data. This is just concentrations. So I guess on that note, I guess we're talking about it right now. We did turn all these concentrations into loads, masses of pollutants based on our treatment volumes, which Chase alluded to. We have area velocity sensors out and pumping rates. So how we did that is we had the event volume from these sensors. So this is showing, this graph is what our output looks like from our area velocity sensors at Woodcrest filter. We have one measuring the total filtered volume, and then we have one measuring all the volume coming off the biochar cell, and that's in gray here. And then the difference of those is all the water that's coming through the iron enhanced cell. So it's not quite as simple as just dividing the total water in half. There's slight differences in the size of these filters and the elevation of the intakes and the drain tile system, because just due to gravity, it has to be sloped. So we're getting more water going through the iron enhanced side because it's a little bit lower in elevation. So what we can do is for every event, just calculate that, the area of water under the curve, and then portion it off appropriately to what filter cell it passed through. And then we apply that event grab sample concentration. As Chase said, just one grab sample per event. We do try to vary it, and sometimes we get the rising limb of the hydrograph or the falling limb or the peak of the storm. So there's some variability there, but we recognize it's not perfect. It's just difficult when dealing with bacteria and when you can't use an auto sampler. But we multiply those to get the event load. And then what we did is we summed up all of those, all of the untreated load, every mass of pollutant that entered our system, and then we summed everything that left each of the cells. And you can compare those numbers to calculate your percent cumulative load reduction of, the year's worth of phosphorus that entered, how much of it did we reduce? We think those are really important numbers to look at for how these things are functioning when it comes to how they're actually, benefiting or not the receiving water. So I'll show you those numbers here. So looking at the Woodcrest filter only, and then I've got the two different filter cells, the B with the biochar and without. When it comes to E. coli, the biochar cell reduced 89% of all of the E. coli that entered it. So that's fantastic. We were shooting for at least 80% and we hit that target. And then the side without the biochar still actually did remarkably well and reduced 72% of the E. coli. And that's, it's expected with a sand filter that you would have just some physical entrainment of bacteria as well. At Pleasure Creek, similar good results, over 80%. The biochar added side had a slight leg up at 87% compared to the 84% of the iron alone. When it comes to phosphorus, looking at Woodcrest, again, we hit our design specs. We were going for 77% removal. That's what the Stormwater manual says for an iron enhanced sand filter. And we hit that. And here the iron enhanced side without the biochar slightly performed better than the biochar side. And I will point out with the design of our filter, they are identical cells, and the biochar side just has biochar added, but we did have to remove some of the iron sand just to make space for that additional volume. So our iron enhanced sand filter side has slightly more iron filings. But really there should be plenty of iron filings of binding sites for several years. So we don't anticipate that being an issue and actually kind of affecting the percent removals in the first few years of data collection here. Looking at Pleasure Creek, we had decent removal of phosphorus, a little bit less than we hoped for, but that's mainly because, as I showed you in those graphs, the incoming concentrations were so low that you can expect, percent removals to be lower. And when looking at that dissolved fraction, we had great removal of dissolved phosphorus at Woodcrest, pretty similar to the total phosphorus results. And then at Pleasure Creek, that's where we did see some leaching. So these might be kind of scary numbers that we're leaching, 10% and 41% of our phosphorus out of the system. But I calculated these into loads, and it's really, really insignificant due to the low concentrations that we're seeing here. So combined in the I guess it was 2.5 months of operation, we only lost 0.1, a tenth of a pound of phosphorus to export. And meanwhile, during that same period, we captured 3.64 pounds of total phosphorus. So we're having a net benefit. There's a little bit of dissolved phosphorus going downstream, but we're capturing more phosphorus. The receiving water wins. And just for fun, I calculated that for E. coli, even though E. coli loading numbers are kind of obscure, but we captured almost 10 billion organisms of E. coli in the first 3 months of operation here. So I thought that was pretty cool. So I did just want to quickly acknowledge that I guess these numbers, this is a very simplified 1-number summary of how this BMP worked in 2020, and that there's a lot more complexity of this, variability between events. So I at least wanted to show some box plots so you can see that variability. So what I'm going to show here is a panel of graphs where, so this is the woodcrest filter. This is just E. coli, and we have the biochar side and the iron side, and these are individual event loads as these bar charts. So here in the untreated water is gray, and then the green is treated water. So you can see that there's higher loads, and then it's significantly and consistently reduced on the biochar side. But there is one outlier. This single event had, almost 6 billion organisms of E. coli. So that highly influenced that 1-number summary that I showed you on the previous slide. Something to point out. So when you look at the iron side, pretty similar, but here the treated water, there's a bit more variability. It's not quite as consistent and quite as high treatment level. So kind of similar story here at Pleasure Creek. And then looking at phosphorus, you really don't see any difference in the distributions between the biochar side or the iron side. But again, there's some outliers that are really driving those cumulative load reductions. And same for orthophosphorus. And here again, you can see that leaching, how this effluent bar, is a little bit larger than the influent bar. But again, all the concentrations are really low. But there was a couple events here where the majority of that export, even though it's 0.1 pounds, it came basically from these 2 events. So with that, before I tell you what happened this year, give you a summary of the 2020 result. Basically, all of our filter cells reduced E. coli and total phosphorus concentrations and loads consistently. The woodcrest filter, that biochar amended side, outperformed the iron side. It removed 17% more E. coli. Whereas at Pleasure Creek, they performed similarly. The biochar side just had a slight leg up, 3% more. The total phosphorus loads were comparable between the different media types. So we had kind of a split pattern. So at Woodcrest, the iron-only side outperformed Biocrest by 5%, but at Pleasure Creek, the biochar side actually worked better than the iron side, so there's no real clear pattern. And then again, for orthophosphorus, at Woodcrest, the side without biochar did outperform the biochar side by 15%, but we had leaching, insignificant amounts of leaching, so it's hard to draw conclusions about the orthophosphorus at Pleasure Creek. So that brings us to 2020. I just wanted to start off with a caveat that this year, as I'm sure most of you know, we had drought conditions, and this did impact the operation and sampling of both of our filters. For Woodcrest, that gravity-fed system, we just didn't have a lot of large events. And at the Watershed District, we have a lot of other sampling that also needs to get done whenever it rains, our stream sampling. So we had a difficult time capturing, the middle of the hydrograph on larger events. And then at our Pleasure Creek system, it's pump based, so we can run it 24/7, but the pond we're pumping out of, due to the drought, it was about 2.5 feet lower than normal. which is right where our intake of our pump is situated. So we ran the risk of drawing down the pond too far and burning up our pump motor. So we kind of had to cycle it on and off and had several weeks of downtime kind of waiting for rain and the pump to rebound. So we didn't get as much treatment as we had hoped that we would get in a normal year. Nevertheless, we have some results. So at the Woodcrest filter, we did sample 10 events. And again, that biochar amended cell outperformed the iron cell, this time by 11%. But our cumulative load reductions were a bit lower. This year, we saw 69% on the biochar side versus 58% in the iron enhanced side. And if you remember from the previous slide in 2020, it was 89-72. And I don't think this is a sign that our filter is already, dropping in performance over time in just the second year. I think it is more an artifact of just more of our poor sampling during the drought condition. Because unlike in 2020, we did see some export of E. coli in some events. And we noticed that these were all kind of the smaller events where we got to the filter late and we were sampling kind of the tail end of the storm. And it seemed like we did have more bacteria coming out of the effluent pipes here versus larger events when the whole filter is flooded and we have a lot of E. coli in the incoming load. So it's just kind of interesting. And I guess we won't know more until the next couple of years to see if we can rebound to 2020 numbers or if we are seeing a real decline in how these BMPs are performing. At Pleasure Creek, we were again plagued by low incoming pollutant levels. So here, only one of our 11 samples even had E. coli concentrations that were greater than that chronic standard. So for this one event, the filter worked great. Both sides, the biochar side reduced E. coli by 98% and the iron side by 99.8%. But the other samples, when you sometimes our influent would be 2 and then you'd have 30 leaving either side. So you have these like negative 800% removals, yet barely any E. coli. It's not meaningful. So we are hesitant to draw any conclusions about 2021 E. coli at Pleasure Creek. When it comes to total phosphorus here, again, we saw great consistent removal at both filters. This is just the same kind of cumulative load reduction numbers. So at Woodcrest again, mid 80% removal, no clear winner between the biochar amended or the iron enhanced side. And at Pleasure Creek, these were lower because of the lower incoming, but we did see between 50 and 60% removal. And here the biochar side again had kind of a slight leg up there. And for orthophosphorus, kind of the same story as last year. We get good removal at Woodcrest, but pretty consistent leaching, consistent but insignificant leaching at Pleasure Creek because of those low incoming pollution. So here we were operating all year and we exported a total of 0.3 pounds of orthophosphorus. But all in all, we captured more total phosphorus. So again, it was a win for the receiving water. So with all that being said, conclusions we can draw is that biochar amendments to sand filter media, it seems to increase your E. coli removal by somewhere between 5 and 20%, and especially when influent concentrations are high, when it would be most beneficial. And that adding biochar to these systems doesn't seem to impact phosphorus removal. It doesn't help it, but it also doesn't hurt it. There were some kind of cautions that adding biochar to these systems would cause phosphorus leaching, much like has been shown with some types of compost and sphagnum moss, but we did not see any evidence of that. So kind of combining those two, it seems like biochar is really a low cost, low risk amendment with great potential to increase the removal of bacteria and also increase other contaminants. So I guess I haven't touched on cost at all yet, but just our two projects looking at our construction costs, if you look at just the cell with biochar versus the cost to construct the cell without the biochar, it was between, in Woodcrest Creek, it was only 2% no, it's the opposite way. In Pleasure Creek, it was 2% more expensive, and in Woodcrest, it was 15% more expensive. So it's really a minimal additional cost. Our average bid price was $220 per cubic yard installed. So that was actually cheaper than just the iron enhanced sand media itself. So if you void some of your volume space and take away some iron sand and add biochar, you can really have a cost-effective addition. And I guess I alluded to this, that some future next steps would be that there's a lot of evidence out there in the literature that biochar reduces other pollutants of concerns, things like heavy metals, pesticides, possibly PAHs. And this is actually something that we would love to do, but it's very expensive to do this kind of monitoring. So we do not have the money for it, but USGS has applied for grants from the state legislature to do this type of work statewide, looking at how our stormwater BMPs are reducing these contaminants of emerging concern. And they are interested in using our sites to test biochar's ability. So we are lending them our sites. So we're hopeful that they get funding and we can have some of these answers in the coming years. And then some new research out of SAFL that's going on right now also shows that adding biochar to your filter media can help support plant growth when you want it. We're not trying to grow plants on our filters, but when you're looking at things like rain gardens, where you're trying to grow plants and more bioengineering practices, biochar could be a way to support that plant growth without getting that leaching of phosphorus. So I guess in summary, it seems like a low cost, low risk with the potential of other benefits too, and kind of modest improvements in how much bacteria we are reducing. So with that, I guess stay tuned. We'll be monitoring these things in years to come and hope to be back presenting in a couple of years. And we would love to take any questions the group has.
We would love to have you come back and tell us more about how this study is turning out. We do have a couple of questions in the chat here. Brett Emmons asks, or starts out by saying that it is nice your side-by-side are the same except for biochar, but do you have a sense that if iron enhanced sand were out of the mix, would biochar perform the same? Would the iron not be needed?
That is an interesting question. We wanted to keep all things even, so we were just looking at biochar's ability, but I have a feeling that it wouldn't. There is some literature that suggests that biochar can remove nutrients, including phosphorus, but we didn't see any evidence that having that biochar gave any extra boost to phosphorus removal. And that's maybe because we selected a biochar that was optimized for removing bacteria. And a lot of times the biochar you use for nutrient removal is burned at a lower temperature. And it's more about kind of active chemical groups and sorbing and less about like surface area and physical entrainment. So I think there's a lot of opportunity there for future research and trying to find these kind of optimal mixes or layered systems, depending on what you're trying to target.
Very good. And Aaron Anderson Wens from Bar asks about on your question slide, what are the rock-like covers shown on the filters?
Oh, yes. So those are just fake rock covers. So we have clean out port, just, I don't know, 6-inch PVC clean out ports for maintenance that were pretty ugly. So we put decorative fake rocks over them. But the local residents refer to them as the graveyard.
Where did you get them?
Do you remember the vendor online?
Cora Rock or something. I can, if they want it, they can email me and I can get it to them.
All right, you can. It sounds like you can email him and he will get that to you. And Eileen was wondering, would you be able to speak more to the future monitoring plan, including the time frame and whether it is being done as part of the grant funding requirement?
Yes, another good one. So we didn't have any explicit requirement in the grant to monitor these, but there was a requirement to create an operations and maintenance plan. And as part of that operations and maintenance plan, we added our monitoring to it. So it's kind of a self-imposed requirement We do kind of a, depending on time, a flexible schedule. So we said for the first 3 years, we're going to do intensive monitoring, 10, 12 grab samples a year, like the data we've been showing. And then after that, it transitions to seasonal, 4 times a year. And then there's some gap years, but we come back the 5th year, 10th, 15th to track kind of long-term maintenance. So that is our plan for now, but we might extend our kind of intensive monitoring period if, depending on, I guess, what we see the year prior, we're flexible on that. Or if we end up working with USGS to track different contaminants, then we might be out there collecting water on their behalf if they're paying for the analysis. So we're open, but we will definitely be monitoring these for the next 15 years.
And Karen Kill from Browns Creek is wondering if you sampled for heavy metals and was there any improvements with the biochar?
We have not yet. So we're hoping to in the future.
And did you cover this? How did you decide to use the 30%? Biochar?
That is a question for Ed Mathieson, our engineer who's not here. I think he started this work working with the Shingle Creek Watershed Management Commission, and they toyed around with a couple of different, between 5% and 50. And I think 30% was just kind of a good compromise where we didn't want to impede water flow, infiltration rates, but have enough of that surface area. So there's nothing magical about 30%. It just seems to be what's working for us so far.
Okay. Any other questions? I think I grabbed all of them in the chat. I'll just, looks like it.
Emily, I had one more.
Okay. Go
And
for it.
It's Justine, can you
there
kind
it
of
is.
give some background? Yeah, it was right afterwards. Can you give some additional background on the grant and permitting process associated with these types of projects?
Yeah. So when it comes to grants, we have been very fortunate in applying for the Clean Water Fund using the competitive program. And because iron enhanced sand is really, kind of a trusted BMP, there's literature supporting it. It's in the Stormwater manual. We were able to lean on that. And I guess I should back up and say, we're not just building these BMPs to reduce bacteria. We're really targeting our conventional pollutants, our TSS and our TP. We are impaired for both of those as well. But kind of our stance as the district is, if we're building these BMPs anyways to treat these conventional pollutants, let's try to optimize these to get bacteria out too. So if it's a harmless addition to pay a little bit extra, put biochar in and increase our E. coli removal, that's what we set out to do. And that's how we spun it kind of in the grant world. So that was not research, that this is a real BMP with the potential of, reducing bacteria and learning more and kind of sharing this knowledge with the rest of the practitioners. And we scored really well. I think, top 3 or something of all the applications in that year for Woodcrest. And then for this Pleasure Creek filter that you see on the screen, that we actually used the watershed-based funding allocation. We pulled our technical advisory committee and just said, we have this extra pot of money. We want to do something big and impactful, and kind of everyone voted to try this out, and that the city of Blaine was willing to put up match $200,000. So that's how this one came to be. And as far as permitting, these did not require any Army Corps or DNR permitting on upland. This one was on upland. So that was pretty easy. And the other one was built on an existing rate control pond. So it was permitted as a kind of a modification to that rate control pond. None of it was public wetland.